Today's lecture is about compound action potentials. Compound action potentials are a subject that we will be using in the ECG lecture, the next lecture. First of all, we have to make a description or the definition of compound action potential. Action potential is a membrane potential change, a depolarization of membrane potential of the cell. However, compound action potential is something that you can record from using an extracellular electrode, and this potential change is due to summation of action potentials occurring in many number of cells. So you usually do not record a compound action potential from a few cells. You usually record an action potential, compound action potential, from many thousands or millions of cells. So an organ usually creates a compound action potential, like heart or brain. Compound action potentials recorded from these organs are usually given specific names like ECG, EEG, so on and so forth. To understand the compound action potential and also to understand the basic theory about ECG, we need to first understand a physical concept called dipole. Dipole is a physical system which has no net charge. However, it can induce an electrical field around it. The reason for that is that the negative and positive charges, which are equal, and their sum is zero. However, these charges in space do not perfectly overlay each other. So there is a gap, or there is a distance between the positive charge and the negative charge in the system. Because they do not overlap each other, this effect creates an electrical field around this physical system, which we call the dipole. An important point here is that the electrical field around this dipole, unlike an electrical field around a single charge, does not only depend on the amount of charge the dipole has, the distance between the dipole and the point you're measuring the electrical field, but it also depends on the angle, which is shown in the picture by theta, which makes a dipole some sort of a vector. So the orientation of the dipole in space, the direction this vector shows, when it changes without changing its magnitude, the amount of charge it has. If you turn the dipole in space, the electrical field you measure in a constant point in space will change. This is an important point we have to keep in mind. So if dipoles are vectors, so you can sum a group of dipoles using the rules of vector summation, and you end up with a compound or summation vector which represents the effect of all these vectors. And you can use this summation vector instead of a bunch of dipoles. The thing is, when you look at a cell which has a membrane potential, you can see it as a collection of many dipoles, as shown in the figure. And so, when you look from a point in space towards the cell, you should have an effect of these dipoles. You can sum these dipoles as vectors, and the net dipole you'll find at the end of this summation process will be the dipole vector, which will have the effect on the point in space that you're measuring the electrical field. However, when you look at the picture, you'll instantly see that these dipoles they are arranged in such a way that they will cancel each other out. For example, when you look at the dipoles which are shaded with pink, and the dipoles which are shaded with blue, the blue dipoles, and also for the pink dipole, the pink shaded dipoles, they seem to have similar, as the potential in the membrane is the same, their magnitude, the dipole magnitude is the same, but their orientation is completely opposite of each other, which when you sum these vectors, you'll definitely see that, or at least you can imagine that, you'll get 0 as a sum. So that these dipoles will cancel each other out. So a cell which has a homogeneous membrane potential will create a 0 net dipole. There will be no net electrical effect around the cell. But however, if you have an action potential, for example, in some part of the cell, of course this is not a stable condition. Action potential will spread through the whole cell, but at this instance in time, this potential, membrane potential of one part of the cell, which is shown here in red, is different than the membrane potential at the other part of the cell. The left side of the cell is different than the right side of the cell. This, you can instantly see that even if the pink shaded dipoles kind of cancel each other out, the blue ones, now they show they have the same orientation with each other, so they'll add up to something. So this cell will have a net dipole, and it will have an effect, electrical field around it. As you can see, this summation depends on geometry, or at least it looks like it depends on geometry. So if we want to understand the situation, for example, what's happening in a long nerve or in a cardiac muscle, we try to understand long cylinder-like geometry. This cylinder-like cell has membrane potential, and there is a point in space outside the cell, which we denote with P. What is the electrical field in this point in space? When you do the math part is a little bit complicated, so I'm not going to explain how you sum these dipoles, but the result is that if you draw an angle at point P, two lines coming out of point P, they will have a field of view which includes some dipoles. The sum of those dipoles, if they have opposite direction to each other, will be 0. This is how the math comes out. It's the same with the other angle. So the closer dipoles has a larger effect, so they cancel out the dipoles which are farther away, but you need more dipoles at the farther end than the closer end. So the math tells us that if those dipoles are within that angle, they will cancel each other out. However, if there is an action potential spreading through this cell, for example, at the right-hand side of the cell, the membrane potential is opposite of the membrane potential on the left-hand side. For example, say the membrane potential is negative 60, 70 millivolts in the left-hand side. It is positive 60 millivolts at the right-hand side. It's spreading through the cell, but we are looking at an instance in time, a second, millisecond in time, and we see that the situation is as shown in the figure. So there are three angles now. On the left-hand side, the cell is polarized, and the dipoles cancel each other out, should cancel each other out, because they have opposite directions, and we know that this from the previous slide that they should cancel each other out. On the right-hand side also, the cell is depolarized. However, again, the dipoles has opposite orientation, so they should cancel each other out. However, in the middle, there's an angle which has within its field of view, the dipoles has a similar orientation, which means that they won't cancel each other out, and they'll add up something other than zero. So this cell has an effect, electrical field, measurable electric field on the point P in space. And again, if you can add dipoles and come up with a summation dipole, which actually can account for or explain the effect, the total effect of all those small dipoles, then this cell has a dipole, which is not zero. In short, if the whole cell is depolarized or the whole cell is polarized, it doesn't matter. If the whole cell, the whole tissue, the cell, has a similar membrane potential, it adds up to a zero dipole. However, if the tissue, the cell, has parts of it depolarized and parts of it polarized, or have different membrane potentials, then you end up with a dipole, net dipole, which means that you can measure an electrical field outside the cell. The important thing is that this dipole, this net dipole, is only created by the junction between the depolarized and polarized part of the cells. The polarized parts and the depolarized parts by themselves do not contribute. As you can see, the theta and the phi do not contribute to this dipole. They add up, the dipoles within their field of view add up to zero. The only part that contributes is the junction between the polarized and depolarized parts of the cell. So the rule is you will end up with a dipole from which is the negative part of the dipole is at the depolarized part of the cell, and the positive part of the dipole will be at the polarized part of the cell. You can see that the depolarized tissue, which is shown in red here, means that it's at the peak of the action potential, but the blue part means that the cells are at resting potential. They have not shown an action potential yet. Their membrane will depolarize also eventually. However, at this instant of time, left part is showing an action potential, the membrane potential is depolarized, or the right-hand part, the blue part, cells are at resting levels, so it hasn't been depolarized yet. You have to understand that the direction of this dipole vector is not the direction of the spread of the action potentials. It's showing the electrical dipole. It's not a movement. Electrical dipoles does not have to move. They may move, they may not move, but that arrow does not indicate a movement. That dipole only shows where the negative part of the dipole is, where the positive part of the dipole is, and what is the orientation of that dipole. Because as we have seen, when this orientation changes, when the dipole turns in space, even if its size does not change, the effect it has on a certain point in space will change. So the orientation of dipole is important. We will use this information in the ECG, so it's essential for us to understand this lecture so that we can understand ECG without much, the theory of ECG without much problem.